Hello everyone, welcome to BioScholars Academy of Learning. So as I had promised, I would like to discuss about the important topics from each unit, and today I would want to discuss about the first unit, that is Unit 1 Biochemistry. So I have just written down for my references, so in case if I forget any topic I could just refer. So the first topic is amino acid. So what is more important in amino acid? The structure of amino acid. Okay, what is the Zwitter ion? What is isoelectric pH? And how do you calculate the charge on an protein? So all that is very important in amino acids topic. So you need to understand what is pK, what is pi, and all those concepts. Next, coming to protein, we are supposed to understand the protein structure, the primary structure, secondary structure, the tertiary structure, and quaternary structure. We are also supposed to understand the two important protein which is included in your syllabus, that is collagen and elastin. Other than this, you should also have an understanding about protein denaturation, protein cleavage. You will have problems based on enzymes like trypsin, chymotrypsin. So they can give you a peptide and they may tell you that we are treating this peptide with trypsin and chymotrypsin, and can you tell me what kind of peptides will be formed, what kind of dipeptide, tripeptides will be formed. You can have such questions. Next, you have motifs and domains. That is also a part of the secondary structure of the protein. And then Ramachandran plot. Something you should not miss while you're studying protein is Ramachandran plot. So it tries to give us an idea about the psi and the phi angle. So you need to understand the peptide bond, what kind of bonds are there, the psi bond, phi bond, the omega bond, all those bonds you need to understand. Next, you have enzymes. Enzymes is very important. So what you need to understand, the principle behind enzyme catalysis, how is an enzyme working, okay, and classification of enzymes based on the type of reaction they catalyze, like oxidoreductases, dehydrogenases, transferases, and all that. That's very important. Then we need to understand the enzyme kinetics, your Michaelis-Menten equation. You need to know what is Vmax, what is Kcat, what is Km, all that. And numericals based on enzymes is again a very important topic. Then again in enzymes you have inhibition. Direct questions from inhibition comes. What, like competitive inhibition, non-competitive, uncompetitive. What is the difference between each competitive inhibitor? What are the examples for these kinds of inhibition? And what happens to Vmax in each case? What happens to Km in each case? And how does the graph change? That is, how does your Michaelis-Menten curve change in case of each kind of inhibition? That is also a very important topic when it comes to enzyme. Other than this, you should also understand the mechanism of enzyme catalysis, like acid base catalysis and all that. Then you should understand what are the cofactor, what are coenzymes. So what is the difference between the cofactor and coenzyme? What are the different cofactors for different enzymes? Like that. Say for example, kinases need Mg2 plus as the cofactor. Like that, you need to study for every enzyme. Okay, and all your dehydrogenases, they require NAD plus as their cofactor, yeah, as their coenzyme. So like this, you need to understand the difference between cofactor and coenzyme, and which enzyme needs what cofactor or what coenzyme. That is very important. Other than this, you have two small topics like allozymes and isozymes, okay, and allosteric enzymes. What are allosteric enzymes, what do they do? That is also very important. So this is about enzymes. Next, carbohydrates. So in carbohydrates, what you should study, again, the types and classification of carbohydrates, like monosaccharides, disaccharides, oligosaccharides, polysaccharides, and all that. And what kind of bond is present between the disaccharides, like what is the bond between your glucose molecules and sucrose, or between the glucose molecules and cellulose, what kind of linkage it is, whether it is alpha linkage or whether it is beta linkage. All that are very important. Okay, yeah. Other than this, metabolism, carbohydrate metabolism is very important. Like your glycolysis, where does glycolysis takes place? What are the different steps involved in glycolysis? What is the net ATP production in glycolysis? And what are the steps which are involved in regulation of glycolysis? Which one will inhibit glycolysis, and which step will, or which product will, or which substance will basically enhance glycolysis? All that is very important. Then after glycolysis, the fate of pyruvate is also important. What is glycolysis? Conversion of glucose to pyruvate. And after that, what happens to pyruvate in aerobic and anaerobic condition is also possible. And it's not possible for me to explain everything here. This video is just to tell what all you need to study from the biochemistry unit. That's it. Then you need to study about Krebs cycle. What is the net energy production in the Krebs cycle? Okay, how many ATPs are produced, how many NADH are produced, how many FADH are produced? All that you need to definitely remember. And what are the regulatory steps, and what are the enzymes involved, and what are the cofactors, which are cofactor or coenzymes which are involved in those enzymes. And that is again a very important topic for asking questions. Okay, then you have your oxidative phosphorylation, ATP synthesis, you have your electron transport chain. All that are very important. Then you have polysaccharides like cellulose and starch, and what is the kind of bond which are present there, what are the monomers which are present in the polysaccharide is also important. Then coming to DNA and RNA, in DNA and RNA you need to understand what is the structure of DNA, or basic, what is nucleoside, what is nucleotide, what are the types of DNA, the aDNA, bDNA and zDNA, what are the differences in their aDNA, bDNA and zDNA, which is right-handed, which is left-handed, all that you need to remember. And then you also need to remember something called linking number. Okay, you will have problems based on linking number. What is a nucleosome, what are histone proteins, and all those concepts. Then you have the structure of the purine and pyrimidine. Okay, structure of thymine, structure of uracil, structure of adenine, guanine. You need to know the basic structure of it. Okay, yes. Other than this, what is more important in unit 1 is interactions. Like what kind of bond, what are the different kinds of bonds: hydrogen bond, van der Waals interaction, covalent bond, ionic bond. Okay, so of all this bond, which is the strongest bond, which is the weakest bond, and more important, what kind of bonds are present in biomolecules. Like for example in DNA. So in DNA the opposite bases are joined by hydrogen bond, but what about the adjacent bases? The adjacent bases are joined by phosphodiester bond, and these kind of questions will come. And if you take for example a nucleotide, what kind of bond is present between the ribose sugar and the nitrogenous base? It's a glycosidic bond. So questions like this can come. So if we keep our basics clear, it will be much easier. And what kind of bond is a peptide bond? Peptide bond is a covalent bond. Okay, like this you will have what is a van der Waal interaction, where all you can see the van der Waal interaction. What is ionic interaction? All this is very important. Other than this, you will also have problems based on pH and buffer. How to calculate pH, how to calculate buffer, the Anderson-Esselbatch equation, all that you need to be thorough with. So other than this, I personally didn't give more importance to fats. In fats, I just studied the beta oxidation pathway and what is the structure of lipids, how is the classification of lipids done, what are phospholipids, what are simple lipids, what are derived lipids, and what are the different names for phospholipids. That I studied, but I didn't give much importance to the lipid metabolism. Okay, but I did study the beta oxidation pathway. The basic thing I just tried to understand. Okay, other than this, you have in amino acid metabolism, you can just study the transamination deamination reaction along with urea cycle. Okay, so if you study all these three concepts, all this much, if you study what all I have told in unit 1, if you study this much, you are almost through 90% of the unit 1 topics. Okay, and if you revise thoroughly, if you revise these topics thoroughly, and if you're perfect with these topics, if you are able to solve all the questions from this topic, then you are through this unit 1. Okay, so I just thought I will make videos for each unit. Other than clubbing all the units and making one single video, I thought I will talk about each and every unit specifically, and how to study these units. And yeah, as I've already mentioned in my previous video, always go for some basic books for understanding the basics, and then go for a lot of high standard books for understanding the higher concepts. Okay, and solve a lot of numericals based on kcat, km, your enzyme number, turnover number, and you have problems based on pH, problems based on your protein cleavage, charge on the pH. So you can, it's a numerical based unit also. You can definitely expect a lot of numericals from this particular unit. Yes, so this is all I had to tell about unit 1 preparation. Stay tuned for our channel. I will be uploading videos uh, very soon. Thank you so much. Do like, share and subscribe to the channel. Keep supporting. Thank you.